Welcome to the Epigenetics Podcast from Motif. Join host Dr. Stefan Dillinger for lively discussions with leading epigenetics researchers. Hear about their past experiments, what they're working on now, and what's coming next. You know their papers, now get to know them and discover the stories behind the science.
Welcome to the Epigenetics Podcast. This episode is a very special one. For one, this is our yearly Christmas episode, and second, this is the first episode where a guest has a second appearance. So thank you, Mitch, for taking the time to be in this episode again.
Glad to be here.
When we talked the last time, which was in February 2024, the ChIP-seq paper was not published yet. It was on bioRxiv, yes, so in the interest of time, we did not talk about it back then. Since it was published in late 2024, and I'm in California now anyway, I thought it could make sense to visit you and talk about this exciting new method. So, the field already has ChIP-seq, cut-and-run, cut-and-tag, and other methods to map protein-DNA interactions genome-wide. So what specific limitations were you trying to overcome when you started developing ChIP-dip?
Yeah, so all of the methods that you mentioned, ChIP-seq, cut-and-run, cut-and-tag, generally work by mapping a single protein to DNA at a time. And they work really well. There have been a lot of innovations even amongst those methods over the years that have really improved resolution, cell numbers, and many other important aspects of the ability to map DNA-protein interactions. But fundamentally, all of these methods work by mapping a single protein at a time. But of course, there are thousands of different regulatory proteins that bind to DNA in different cell types and different combinations in different locations that ultimately control and orchestrate gene expression. And so mapping a single protein is really only a tiny, tiny portion of the entire picture. And so what we were really hoping to do is change that landscape, go from the ability to map a single protein at a time to being able to map many regulatory proteins at a time, to really get a more comprehensive picture of the regulatory landscape of chromatin and of gene regulation.
So can you talk about the timeline of the genesis of this method? How long did it take for you or your team to make it robust and reliable?
Yeah, so it took several years, to be honest, to go from the initial idea to even the initial implementation to really making it work reliably and broadly across many different protein types and at the scales that we ultimately wanted to achieve and ultimately did achieve. I think the initial development of this was something that we started working on. My student, Andrew Perez and Isabel Garanzi, started working on in about late 2021. And so really over the next several years, not only did they take the idea and reduce it to practice, but really worked out many of the technical aspects that were important for ensuring the breadth and reliability, robustness and sensitivity and specificity of this approach.
So I guess there were also some setbacks in the process?
Well, just a lot of learning experiences, right? Obviously, ChIP itself is a robust method. It's been around for a long time. But when trying to incorporate this into a new strategy like ChIP-dip, which integrates, we could talk more about this later, but integrates many different technical components, including split pool barcoding, being able to link up antibodies and oligos in a reliable and robust way, that really required systematically testing many different aspects of what could go wrong and how to reduce that from happening. So, for example, a normal ChIP experiment, if your antibody and your chromatin dissociate through the process simply because of the binding affinity of your antibody to its epitope, that may not be a major issue because, worst case scenario, you just lose that piece of DNA and you don't see it. However, in a ChIP-dip experiment, that could potentially be problematic if that piece of chromatin, for example, dissociates and then rebinds to another bead, even if it's of the correct antibody type, because now you will no longer preserve the entire barcode. And so that chromatin is no longer something that you can actually map reliably. And the same is true for many of the other aspects of the system, right? So there are many additional potential failure points that could lead to both reduced sensitivity and perhaps even worse, reduced specificity when if not properly addressed. And so those were the kinds of things that that they really systematically enumerated, thought through, and really tested, and in some cases identified issues that they ultimately were able to come up with clever solutions to address and to resolve.
You already went into the details of the problems, but I guess it would be good if you could just give us a walkthrough through the ChIP-dip workflow.
Sure, absolutely. So maybe let's start by just so everyone's on the same page with just the basic workflow of a ChIP experiment, which is that you take an antibody that would bind to your protein, your protein of interest, bind it to, for example, a resin like a magnetic bead, and then use that to immunoprecipitate a protein bound to cross-linked to DNA. And then we sequence the DNA that's associated with that protein. Now, usually we do that one at a time, right? We prepare beads with antibodies. We take cross-linked cells, sonicate or otherwise digest DNA, capture, sequence. But for ChIP-dip, what we do that. But what we want to do is actually expand the number of different antibodies we can utilize at the same time. And so what we do, instead of using a single antibody bead mix, we actually conjugate different antibodies to different beads individually. And we barcode these beads using a specific oligonucleotide sequence, so a biotinylated single-stranded DNA molecule that has a unique barcode that tells us what the antibody is on that bead. And we then take these bead oligo antibody conjugates, mix them together, and basically use this pool of beads and antibodies as our to perform our immunoprecipitation reaction. So just like we would do immunoprecipitation for a traditional ChIP experiment with beads, with a pool of beads and a single antibody, here we would use a pool of beads with this mixture of different antibodies, where the oligo effectively demarcates the identity of the bead type. And the reason why this is valuable is because if we can actually do this and we can resolve the DNA that's bound to an individual bead, we can effectively treat each bead like an individual reaction. And that is that we can do an immunoprecipitation, we can bind proteins to their antibodies and their individual beads on individual antibodies, and then we can do a split pool barcoding to add individual tags sequentially to the chromatin DNA, as well as to the oligo that is on the bead. And so what happens is that the way I think about it is, if you were to take this material and you were divided up into 96 different wells with 96 different tags, then the probability that two molecules, that is a chromatin molecule and the bead oligo, that are on the same bead, right, that are linked together because they've annealed through the antibody, get the same tag is 1. But the probability that two molecules that are not on the same bead get the same barcode is 1 out of 96. And I'm just using this for illustration purposes. We actually don't do 96, but the math is the same. But in fact, I'll just say 100 for the sake of simplicity of the math rather than 96. But and so the probabilities are better that you'll actually share a barcode if you're on the same bead. But of course, we have millions of beads, right, and millions of chromatin molecules in this reaction. So 1 out of 96, or 1 out of 100 in my updated numbers, are actually not that great. We still can't resolve pairing. But what we can do is we can actually iterate this. And so we can then pull things back together, split them again, and can categorize these tags. And so instead of, we'll use a 100 number, instead of 1 out of 100, now we do it again, it becomes 1 out of 100 squared, right? And we can then pull it again, do it again, right? It becomes 100 cubed. And so the probability that molecules on the same bead get the same series of tags across all rounds of this process is still 1, because they stay bound, so they always travel together through this split process. But the probability that two molecules that are not on the same bead get the same barcode, right, decreases exponentially, so much so that we can ensure that the probability of what I'll call a collision, that is, two molecules that are distinct, not on the same bead, get the same barcode, is very unlikely, is vastly lower than the total number of molecules and beads in our sample. And that then allows us to then match up, to then sequence, right, the sequence of tags, which we call a barcode, that are on the oligo and that are on the chromatin, and match them up. So that is that if they have the same barcode sequence, they must have been on the same bead. And so we can now resolve the identity of all these different proteins and the DNA that's bound to it by simply assigning these barcodes together into clusters. And so the effective output is the same as a ChIP-seq experiment, except instead of getting one map, we can now get hundreds of maps.
So what is the difference of the different wells?
So each well in the split pool barcode would have a different sequence. So it would be, you would
have a different bead in each of the wells?
No. So the beads, it's the same beads. The beads are going, you basically are pipetting into different wells. Each well has a barcoded tag. So a DNA sequence that we ligate to the oligo to the chromatin that has, for example, one of 96 different sequences. And in this example, right? So it could have, sequence ACGT, and the other one could have sequence GGGC, and the other one could have sequence CAGA, something that we can identify uniquely in the sequence itself. And then what we can do is we have their units. So each tag, is a defined unit we can identify, we can map, and we can match. And what we're looking for is basically matching, all N units, right? In our case, I believe we do 6 rounds. So it's basically 6 units that we're matching across.
And you would have all antibodies in one well?
So all antibodies will be on beads within one tube, yes.
Okay.
And so we will do that. We do the IP first in the pool of antibodies. And then after the IP, we dilute the material into these wells, add these barcodes, right? Pull it back together, split it again, and add these barcodes. And so it's that step that allows us to resolve them.
Yeah, because each individual antibody chromatin particle would then be randomly assigned to those wells, and then this is why you get the individual barcodes for each of those.
Exactly. One way that I often think about this split pool process that may be helpful is, imagine that we were able to do this same experiment where we put a single bead with a single antibody type, and we were then able to, come up with some really clever microfluidic device that can isolate that individual bead and that individual chromatin, right? And we can just uniquely tag that isolated bead, right? We would be able to tell all the chromatin that was in this isolated microfluidic chamber with this antibody type is for this protein. It binds to this protein. But that's hard, right? There are many, many technical challenges there that make that not feasible. But what split pool allows us to do is effectively mimic the ability to isolate and define individual beads so that we can map the composition of each bead uniquely.
So I don't know how long those tags on the sequence will be then in the end. So traditionally you would sequence like cut and run, cut and tag and things like that with PE 35, whatever it is.
Yeah.
So does this change because the barcodes get longer maybe?
It does, yes. And actually, for all of these split pool methods that we've been developing over the years, that is, I would say, I don't know if it's a limitation or not, but certainly one of the limitations, if you will, is that you do need longer paired end reads. But generally, the length of the reads that you need, it can be asymmetric, but the longer reads are on read 2, the second of the reads where you're reading the barcode itself. The chromatin, you can, of course, read at whatever lengths, single end or paired end that are standard for, different people's workflows.
Can you maybe summarize the critical step that makes the method unique? Is there only one, or would you say it's more than one?
I think the key step that makes this unique is the split is basically resolving these multiple proteins in the same reaction using split pool barcoding. Although I guess really now that I reflect on this, there are really 2 fundamental unique aspects of this that work together. The first is the ability to barcode bead antibody conjugates, right? To label all of these different bead types, right? In this case, bead types being beads bound to different antibodies. And the second is being able to do a pooled ChIP. And the second is to be able to resolve each of those beads to individual chromatin molecules. Because, of course, doing a pooled chip in and of itself doesn't really buy you much. It just buys you a big cacophony mess unless you can resolve it. And that's where the split pool provides a lot of value is in being able to resolve them.
So how sensitive is ChIP-dip to antibody quality? And how did you validate that the method faithfully captures the intended protein-DNA interactions?
Right. So there are, I guess, two components to that. I mean, the first is the standard issues with antibody quality also apply for ChIP-dip, right? You have to have a high-quality antibody to be able to purify the protein. There's an antibody crisis, if you will, in the biomedical sciences. We haven't solved that, and that's obviously something that we too are sensitive to. If there are certain proteins for which there are no high-quality antibodies, you can't, at the moment, can't do ChIP-dip on it. And that's a standard problem that we all unfortunately share. But there's a second, I think, unique aspect to ChIP-dip, that potential aspect about quality that affects ChIP-dip that is different, and that is, thinking about the pooling aspect. You can imagine that if you're doing a single chip and you happen to pick an antibody that doesn't work or doesn't work well, that the worst case scenario is your experiment doesn't work. But if you're doing a pool of, let's say, 100 different antibodies and you happen to pick one that doesn't work well, will a bad antibody spoil the pot? And that is a unique challenge for thinking about ChIP-dip or multiplex methods in general that are an additional challenge beyond just the traditional antibody limitations. And that's something that we spent a lot of time, worrying about, a lot of time exploring and really trying to address and resolve. And maybe in the interest of shortcutting a lot of the work we did on those fronts, what we found is that, was that that's not the case. That even if we have antibodies that we include in our pools that either don't perform well or even that have high levels of background binding, that yes, it leads to the inability to map that protein with that antibody, but it doesn't impact the quality of all the other data and all the other antibodies in the pool. And not only is this, of course, reassuring because of it means that you don't have to, you can pool components without having to worry about, will one component mess up my whole experiment? But it also allows us it also becomes a real benefit in many ways, because we can start to do things like utilizing multiple different antibodies against the same epitope. So, for example, we can use many different antibodies against, for example, CTCF or, some other, any transcription factor, chromatin protein of interest, which may include antibodies that may not have previously been validated for ChIP. Not that, which include, right, in fact, many antibodies that we know can work well for other applications have simply not been tested for ChIP. But we can include them as in our pools as a way of effectively testing whether or not these are ChIP-grade antibodies. And so it becomes a way to screen antibodies, but it also becomes a way to build consensus maps. Because if we see that 3 different antibodies against the same epitope give us similar patterns, we can validate that, in fact, this is likely a specific map for this protein. And that's been a really powerful approach. In fact, going back to sort of the traditional challenge around antibodies, the gold standard way to validate antibodies is often to use multiple orthogonal antibodies and identify concordance and consensus in their profiles. And this is just practically challenging, especially when you're doing things one by one, because it just explodes the numbers and the costs. But with a method like ChIP-dip, that begins to become, a very practical approach to start thinking about validation and robustness.
So if it's a target with a very low abundance, you could, because it has been done in the past, maybe fix the antibody to chromatin, because I think if you pool your things again, you pool, you mix, the antibody might fall off chromatin again. Right. So did you test like something like that to fix after the IP?
Yeah, we actually have thought a lot about that and thought about whether that would be something that we needed to adopt. In practice, we haven't, and because we haven't needed it, in fact. and that's prime. Okay, so certainly there are probably some antibodies where that could be helpful. We haven't found that to be necessary for most antibodies we've tested, and we haven't done it primarily because when we don't need it, that of course can potentially introduce other sorts of background issues into the procedure. But the potential for chromatin to dissociate and rebind, of course, is problematic. I mean, for all assays, but particularly for an assay like ChIP-dip, because in a normal chip, if chromatin dissociates and then rebinds to the same antibody on bead, it doesn't really matter because you're still pulling it down. Yeah. In ChIP-dip, if it dissociates and reassociates with another bead of the same antibody, the barcodes get scrambled, so we can't identify it. And we tested this extensively to see whether or not that is the case through several different clever experiments that Drew and Isabel designed, leveraging human mouse sort of mixing experiments, and we just haven't found that to be a major problem, at least for most antibodies.
You also tested on the one hand how many proteins or factors you could analyze in one sample, and on the other hand, how low you could go in cell numbers. So can you comment on both variables in a ChIP-dip experiment?
Sure. So let's start with cell numbers. So obviously, cell numbers are an important consideration and frankly, an important limitation in methods like ChIP. In fact, I think probably the number one critique of methods like ChIP versus cut and tag and cut and run are, the relatively high numbers of cells that you generally and historically need to do a ChIP experiment. And this was something that, we too were, very sensitive about when we were thinking and about ChIP-dip and developing ChIP-dip. But there were a couple of things that we considered here. One of them was that, even with larger numbers of overall cells, if we could map, let's say 100 proteins with the standard amount of cells that you would use for a ChIP, that then effectively the number of cells per protein that you need is reduced by a factor of 100. Because rather than, let's just say, needing 1 million cells for each protein, you need 1 million cells for 100 proteins, right? That reduces the number of cells that you need to, on the order of about 10,000 cells per protein. And so, even that alone, right, becomes a, really valuable proposition in terms of thinking about total cell numbers and access. But in practice, we've also found that by being able to map many proteins together, we can also effectively reduce the number of cells, the total number of cells that we can use going into a ChIP-dip experiment relative to an individual ChIP experiment. And it's hard to say we know the exact answer for why that is, but we think the reason for that is for two general reasons. The first is that the major limitation in the number of cells that you need for a ChIP experiment is really about the amount of DNA you're pulling down. So, for example, if I took 1,000 cells and I tried to do a ChIP experiment on 1,000 cells, right? You have some background DNA that that sticks to your tubes, to your pipettes, to your beads, and then you have signal. And what you need is your signal to be higher than your background, and your background level is going to be some fixed amount, and your signal, will be relatively low because you have very few cells in there, and the actual target DNA sites are a very small fraction of the total DNA. And so it's often very hard to kind of clearly separate the signal from noise when starting with small amounts. But if you can now pull down 100 different proteins, so the amount of signal that you're pulling down is actually is much higher. Let's even say 100x higher, right? And the background level is the same, because the backgrounds, the DNA that's sticking to tubes and to tips and to beads, right, are the same whether you're doing 100 proteins mixed together, the antibodies and beads versus one. You can increase the sensitivity in detecting individual sequences and individual proteins. And so in practice, we've actually found that we can reduce the overall amount, the total number of cells that go into a ChIP-dip experiment, and still generate high quality maps. So for example, in our paper, we showed that if you just took the same pools, about 50 proteins, map it with 50 million cells, 5 million cells, all the way down to 50,000 total cells, which corresponds to about 1,000 cells per protein, we can get highly comparable and high quality ChIP maps for those proteins, even though the idea of generating a ChIP-seq data set for 1,000 cells for any of those individual proteins, I think is squarely, of sufficient quality, I think is squarely in the realm at the moment of science fiction.
I always wonder, where is the cutoff when you would lose peaks?
Right.
In terms of number of peaks,
absolutely,
right?
Yeah. And I think that's the right question. I mean, I think there's the sensitivity question, there's the specificity question. I think, specificity is there. Sensitivity, of course, also depends very much on just the amount, the amount of DNA that it binds. And so, certain proteins that bind to a lot of different DNA sites, you will probably, we know you can get, for example, really, you can get similar numbers of peaks even at very, very low cell numbers. For lower abundance proteins, like transcription factors that bind to fewer sites, even though your specificity might still be there at lower numbers, you will start to lose your ability to detect many individual sites because simply you don't have enough of those binding sites to measure. And that depends very much on the individual protein type. But in our pools, we have seen that, for example, certain chromatin regulators or transcription factors, if you look at the raw tracks, the signal heights, if you will, certainly decrease as you decrease the total number of cells, even though the specificity at those sites stay the same.
Oh, that's good to know. So you now talk about 100 proteins, factors per ChIP-tip experiments, right? And this hints that you could really get, like, I don't know what the right term would be, like a interactome, connectome, whatever it is. So what is going on in the cell on an epigenetic level. So what you're looking at classically is enhancer regions, transcribed regions, promoter regions, polycomb expressed regions or marked regions, constitutive heterochromatin regions, and then of course the transcription factors that go with it. So there is like histone marks that would be an indication for all of those, and you could of course do all of that, but what would you consider like a minimal or maximal set of factors to get like a complete picture of the regulatory chromatin landscape?
Yeah, that's a really great question. I don't know that there's a single answer to that question, to be honest, because it really depends a lot on what, if you will, what granularity you're looking at and really trying to understand. For example, if you really just want to annotate different types of regulatory elements, genes, promoters, enhancers, there are a minimal number of histone modifications that you can do to do that. And for example, K27 acetylation, K4 trimethylation, K4 monomethylation, you get enhancers, promoters, K36 trimethylation, you get genes. If you want to understand beyond just whether or not something's an enhancer, but also its activity state, like whether it's an active enhancer or repressed enhancer, or if you want to understand even more than that, the type of activation, the type of activity or regulation of a given enhancer. So, for example, in our paper, we were able to define different acetylation signatures that define not just activity, but, for example, enhancers that are stimulus dependent versus ones that are developmentally regulated versus ones that are poised for activation. You may need many more acetylation marks. In fact, one of the really, I think, intriguing observations that we found were that if you look at just at a, if you will, sort of a binary level, where is acetylation? All enhancers have all of these different, dozens of different acetylation marks. But when you look at it at a quantitative level, they're actually very different. Different types of enhancers have very different acetylation signatures, and they can define very different types of functional activity at these different enhancers. And so if you're interested in that kind of detail, right, you might need to be able to look not just at a single acetylation mark, but at for example, a dozen different acetylation marks. Similarly, right, there are different types of activity states for promoters. And rather than looking just at, for example, K4 trimethylation, right, being able to incorporate different acetylation marks as well as K4 trimethylation can really define that granularity Now, of course, that's just histone modifications. At the level of transcriptional control, what really drives activity of gene expression, not just what genes are on or off, but at what levels a gene is transcribed, really fundamentally comes down to a combinatorial control problem of what transcription factors are there and what combinations at what sites. And we certainly don't really understand that at any real predictive level. That's been, I would say, a longstanding goal of molecular biology for decades. But a big part of the reason why that's been challenging to answer is because we don't really know what factors bind where, at what levels, in what combinations, and their relationships to transcriptional output. And I think at that level of understanding, it may really require us to be able to map many, many different, dare I say even, hundreds of different transcription factors simultaneously in different contexts.
So exactly what ENCODE did like 20 years ago.
Yeah. Well, I think of, ENCODE was an important project in really starting to look at many of these combinations of activity states, both at chromatin modifiers, transcription factors. The challenge, of course, is that it took a consortium to do that, and they could only and a lot of money, right? Hundreds of millions of dollars were invested in those programs across dozens of labs across the world, and the data has been really important. We use it all the time. I'm sure thousands of labs across the world use it probably daily, but fundamentally these are maps that exist in a few reference cell lines, mostly cell lines, human cell lines, because of the fact that was what was easier to generate these data with. But unlike a genome sequence where, having a reference map can be transferable, at least at some level, to other, for example, to other individuals, to other contexts, a reference cell map for transcription factors chromatin is more limited because fundamentally every cell is different, right? And the epigenetic state, the transcription factor states are entirely distinct between, a K562 cell, a HEX cell, and frankly, a brain cell or even a specific type of neuron in the brain. And so this is where the idea that we would do, something like an ENCODE style project to really understand this entire pattern of protein binding, of transcription factor binding, chromatin regulator binding, histone modifications across all these different cell types really isn't possible when we'll say in the previous paradigm of this one at a time strategy. And I think that's where ultimately what we were really hoping to try to address is if we can generate not reference maps, but allow individual labs to really explore these dynamic profiles in their systems of interest, in their genetic perturbations, in their disease models, in their cell types, in their model organisms, right? That would enable, a real detailed exploration of gene regulation at a whole nother level.
Would you consider this like the method's strength? And what do you think could it then do in the future if you combine all those factors?
Yeah, I mean, I certainly think that's the strength. I mean, my hope would be that this is a first, but still only a first, but a first step towards really, I'll say, democratizing, but really providing the ability for individual labs, obviously our lab, but many others around the world, to be able to start asking the questions that are of most interest to them in their systems, unconstrained by sort of the technical limitations, the financial limitations, and the other practical limitations of the technology, of the mapping technologies. There was a second part to your question that I now forgot, that I missed. So is this the strength? And then what was
the future?
And for the future. Yeah. So, as I said, I think this is the first step. I think that to really maybe fully realize that vision, there are probably several different additional components that will be needed. One of which, of course, is the analytical one. I mean, I think not only did ENCODE have dozens of different labs generating the data, but they also had dozens of different labs analyzing the data, right? Developing computational tools to integrate all this information, right? Some of the, we'll say greatest minds in the computational biology were working on this problem. And that becomes a whole different scale of challenge when individual labs can generate that scale of data in a week versus over many years. And so I think the computational tools to be able to obviously analyze this, visualize it, understand what it means are going to be important to really enable that.
Maybe we can come back to the analysis part later, because I want to come back to the cell number question.
Sure.
So maybe people in the beginning won't do like 100 antibodies, but maybe 20 to 30. And maybe you can comment on what range of cell number they would need in that case. And also, what are the limits then when you look at sample types, right? I mean, we are talking about cell lines.
Sure.
In this case, but is it also like applicable to like tissue or cancer samples where you would have really limited amounts?
Right. Yeah. So I think the answer in terms of the number of cells ultimately will probably depend very much on what the nature of the pool is. So, 20 to 30 histone modifications, I would imagine one can even go lower than 50,000 total cells and get high quality maps. 20 to 30 rare low abundance transcription vectors, probably in reality, you'd probably want more than 50,000 total cells to be able to get really high quality maps, maybe on the order of 1 million total cells. And, that may also depend on the cell type and the specifics of the cell types themselves that obviously may vary. Will that be a limitation? I don't think so. Obviously, I'm not an expert in every cell system. And so I'm sure there are cell systems where truly getting 1 million total cells is completely infeasible. But the way I think about this is, I'll use it by an example from one of my colleagues that we've worked with here at Caltech, who works on these primary immune cells, very early development of primary T cells, and isolating these early developmental states are really challenging, and it's very because they have to come from a very, very specific fraction of the mouse blood, and they're a tiny, tiny proportion. So, in the history of studying this system for decades, they've generated, ChIP maps for, 1 or 2 histone modifications. I think it was K4 trimethylation, K27 acetylation. Through a lot of work. What's that?
The most abundant ones.
The most abundant ones. And so, getting the cell numbers needed for that, was work, but it was doable. And so, it's possible. The idea of doing that doing that 20, 30 times, that would be crazy. But if we could, take that same, let's say, 1 million cells or whatever number it was that went into their chip and do that across the 2030, right? And to have a comprehensive profile, that entire developmental state from that one collection, that is not only doable, but incredibly valuable from a data perspective. So yes, often the idea that of collecting these large numbers of cells becomes very limiting when you think about it across the many, many different conditions that you might need to map to be able to fully understand that rare population or that cell type. But by amortizing that across the number of proteins, even if it's a rare population that's a lot of work to get, may be possible and even valuable if you can actually generate these, comprehensive profiles. And that's where, larger numbers, larger pools may actually even be more valuable, not just in getting more data, but potentially even in reducing the total number of total cells that one needs to be able to get high quality maps.
So I just thought of something else. So what happens if, there are many modifications on the same nucleosome, but is it possible that like a bead or an antibody binds to the same nucleosome, and what happens in that case?
Yeah, so that's a great question, and actually one that we thought a lot about, not as a problem, but actually as an opportunity. because in a way, if you were to get multiple beads binding to the same nucleosome, then not only would we be able to map them, but we would actually be able to observe co-occurrences on the same molecule of chromatin. In practice, we don't see that.
Okay.
And primarily, the reason we don't see that, we think, is because of the sterics of the beads themselves. The beads are about a micron in diameter. Simply getting multiple of them to bind on the same chromatin fragment just seems to be sterically not very uncommon. And so that doesn't seem to be the case.
So coming back to the amount of antibodies, in the discussion part of the paper, you discussed that you can generate datasets with pools containing more than 160 distinct antibodies. I mean, it won't be possible in a 96 well plate then. But I mean, this is great, but is it also necessary, especially in the light of data analysis and maybe also limited funding available in labs, because 160 antibodies would be...
Sure. Yeah. I mean, sure. Of course, those are real practical considerations. And of course, the cost of individual antibodies is, well, frankly, obscene. And understandable reasons why, but still, right, the idea that, you would buy 100 antibodies from a company is probably out of the scope for most labs. Honestly, including our own lab. I mean, we were lucky that we were able to get access to antibodies from companies like Motif and Cell Signaling and others that were, generous in providing us access to this. But of course, ultimately, that's not necessarily going to be the case for every individual lab. So, we're certainly understand that limitation. My hope would be, of course, that with the adoption of these pooled-based approaches, rather than having to buy, 100 different antibodies or even 50 different antibodies or 30 different antibodies, that companies like Motif or others will actually fully sell pools of antibodies for these kinds of purposes that are, more economical for individual labs to buy and use, rather than having to buy each individual antibody on their own. And so from that perspective, I'm hopeful that there can be a new business model, perhaps, that would benefit, we'll say, antibody companies as well as individual labs by changing sort of how the unit, if you will, is packaged, sold, and produced. Now, having said that, right, of course, the cost issue is not just about, having the antibodies. It also is about, the amount of data you need to generate. For every read you're distributing across 100 proteins, it means you need more reads to get higher coverage for each protein. And that's, of course, a real consideration. I think the cost of sequencing has certainly fallen enough that I don't think of that as the major cost bottleneck at the moment, but it's certainly not an insignificant consideration for many. And that's, of course, a trade-off, I think, that every lab needs to navigate in terms of what is the question of interest that they want to pursue, what's the information that would allow them to tackle that question, and sort of what that right unit, both in terms of numbers, but also in terms of the types of proteins that they're looking at that are needed and make sense.
So in cut and run, you would need less antibody as in a traditional chip experiment, because you would need like 4 micrograms or whatever in a traditional chip experiment. So how much antibody would you need in a chip-dip experiment?
Yeah, that's a really great question. And I can't give you an answer off the top of my head. What I will say is I would have to actually go back to the methods of the paper to remember how much we used. I mean, I think when we first started, we actually did start by using a similar amount of antibody as we would for a traditional chip experiment for each component, but that is certainly overkill. And what we know is a couple of things. The first is that a lot of it depends on also the abundance of the protein target. So, for example, for very abundant proteins, you actually want to use less antibody than for more abundant proteins. And we describe some simple ways to actually normalize these antibody abundances, we'll call it the bead concentrations per protein types, to balance those, both at the level of balancing the amount of antibody you're using, but also to increase the yield and balance the composition of the outputs, the chromatin outputs that one generates. We also know that using more antibody, if you will, can not only be more expensive, but also can lead to can also be problematic in other ways. So it's not that it would increase the signal, it would increase the noise level. But for example, if you have a lot of antibody, let's just say you have a lot of antibodies in beads, then the number of chromatin molecules per bead will be very low. So for example, if most of my antibodies, if I have huge excess of antibodies, most of them are not binding to a target, that would mean that on an individual bead, I might have one chromatin molecule or even no chromatin molecules, right, that are actually bound, which means that I need to map that bead and that oligo for no information on the chromatin. So that means that I need to sequence far more of my oligo than my chromatin. That's not a great trade-off because you're just wasting reads on uninformative things. On the other hand, right, if you have lower overall bead density and antibody concentration per target, right, you can drive many chromatin molecules on a single bead. And these beads are big, so they can actually accommodate many chromatin molecules. And so that means that if you have, for example, 10 or 100 chromatin molecules on an individual bead, then for every one oligo that you have to map by sequencing, you map 100 chromatin molecules. And so those are also very practical benefits, not just for reducing the cost of antibodies that you need to use, but also in reducing the cost of sequencing and increasing the information content of the experiment itself.
So in your initial paper, the ChIP-dip assay was performed on bulk samples, not single cells. So do you see this currently as a limitation?
I mean, yes and no. I mean, certainly it is a limitation. I think the real question is, for what specific questions and what specific goals. I think for most applications, ensemble mapping has proven to be very informative and will remain so. I mean, most of the maps that have been generated, for example, historically by ChIP, even by cut and tag, cut and run, are ensembles. Most, of the samples and most of the maps generated by ENCODE are in ensembles. But yet, even in those ensemble maps that we've been able to learn a lot from, we're missing most proteins, most regulatory proteins, and certainly most cell types and contexts. So I think there's certainly still a lot to be learned, even in the ensemble context. Nonetheless, there are, I think, very specific contexts where, it may matter a lot. For example, in very heterogeneous populations, if you were to take mixtures, for example, of blood samples of PBMCs, for example, which are mixtures of many different cell types, and you just did an ensemble, ChIP or ChIP-dip experiment on it, you'd be averaging together the profiles of many different cell types. And that would, of course, be a limitation if you're trying to understand, the regulatory programs of individual cell types, or even, frankly, what are the regulatory programs in any given cell, because, of course, averaging can, lead to ensemble profiles that may not be representative of any cell type. And that is a limitation, and it's a limitation, I think, of all ensemble methods. In addition to sort of just we'll say the technical limitation there, I think there are also, real opportunities in terms of understanding gene regulation that come from being able to generate these types of maps in single cells. And that includes really being able to understand, even within non-heterogeneous populations, the combinatorial dynamics of how multiple factors bind in and relative to each other in a single cell. And those are real, they're not overcoming perhaps the technical limitations as much as providing new opportunities for really being able to understand gene regulation that I think single cell methods would allow us to explore. And so we're not there yet, but we have been working on methods that would allow us to actually generate, to basically adapt and utilize ChIP-dip in single cells. And hopefully that will be something that we'll have available in the near future.
So it's pretty common these days to integrate 2 or 3 modalities or data sets. So I'm thinking of ATAC, RNA-seq, and then maybe integrating something else like ChIP. I don't think that data integration with 10, 20, or 50 data sets will work using the same types of pipelines or strategies that we used with doing this. So what kind of data analysis approaches will need to be employed to integrate these large numbers of data sets, to capture. I mean, it's large data sets, right? So you have like 60 tracks above each other,
Right.
and capturing like patterns will be very hard.
Yeah. So I think I would say there are two parts to this question I'll answer. I mean, the first one is the question of integrating multiple data sets. And I'll disagree with maybe the premise of your question that it's not feasible to integrate multiple different data types using these types of measurements. I would actually go as far as to say that I actually think we can and we have actually integrated ChIP-seq with, for example, spatial, like 3D mapping, sort of our own methods called Sprite, but are very similar to methods like Hi-C that we can, in a method we've been calling CIP-dip, Sprite with IP done in parallel, where we can map, for example, many different proteins using the same ChIP-dip approach, as well as the spatial 3D interaction of DNA. We've also, can incorporate this with, for example, RNA-seq transcription levels by mapping RNA within the same chromatin, on the same beads, on the same chromatin contexts as these. And fundamentally, and we described this a little bit in the discussion of our paper, but really fundamentally what this, as well as the single cell question you asked me earlier, ties into is that ChIP-dip isn't, I don't think of it as a method in isolation. I think of it as sort of a tool within a larger toolkit of these split pool methods that my lab and others have been developing over the last few years, and that includes methods that we've developed for being able to use split pool methods for doing single cell DNA mapping, split pool methods which we published a few years back for 3D mapping, split pool methods for being able to do RNA and DNA simultaneously, which we published a few years ago for. And I think we talked about last time on the podcast for looking at non-coding RNA localization in space, and really, integrating this into a single, we'll call it modular structure that would allow us to sort of integrate and plug and play these different components together. So I do think that it is possible. Having said that, the computational challenges are going to still be there, and whether it's integrating with other existing data sets or ones that are done simultaneously, the scale itself provides a challenge, and that's absolutely true. And often that challenge is, well, going to probably now be the big bottleneck for most labs, right? If the experimental bottleneck, if we've resolved the experimental bottleneck, now the computational bottleneck is going to be the one that I think most will face. And on the positive side, I think that a lot of the developments over the last few years, well, both between developments, computational tools that the ENCODE project has developed for looking even at reference maps are transferable for thinking about any type of map. So, for example, methods like ChromaHMM, this chromatin hidden Markov model developed by Jason Ernst and Manolis Kellis and others, that allow you to integrate different histone modifications into states. That's an approach that we ourselves used in the ChIP-dip paper, but can be applied to any cell type if you have these types of maps, and will be already accessible from a lot of work that they and others have done, even on the ENCODE datasets and these reference maps. I think there are also sort of whole new types of methods that will need to be developed, and hopefully the community will begin to develop software packages that will be accessible for others to use and explore, from how to visualize the data, how to identify patterns, how to extract meaning, and frankly, how to interpret. And I can imagine scenarios where a lot of the recent advances in large language models and AI can actually be really valuable in providing new interfaces and new approaches for scientists to actually explore such complex data sets. Perhaps even for training such large models, for being able to derive new insights and new understandings that frankly have not been possible, in large part because we've lacked the large data sets needed to train these models, right? I like to think about, what has enabled this explosion in large language models like ChatGPT. And yes, there were, huge, big innovations in the computational methods, like development of things like transformer models. But fundamentally, all of that's important, but without the ability to train on the massive amounts of text-based data that was out there, right? These models themselves wouldn't have been able to do much at all. And I think that's, where we are in a way right now in genomics and biology. Many of the methods are out there, in part because they've been developed for a lot of these other applications, computational methods, that is. What we're lacking are really a lot of the fundamental data sets, large data sets to train on. And if that becomes possible, if labs are generating such data across different systems, models, cell types, even organisms, I can imagine, that would enable a next generation of training parameters that would allow us to really understand biology at a whole new level.
So one final thing that we did not talk about yet is the everlasting question of normalization. Because it's done in the individual assays, right? But if I understood the paper correctly, this would not be necessary here because you do it all in one batch.
Yeah, I mean, I think that certainly, the ability to map many different proteins in the same experiment removes a lot of the experimental variability that is involved in many assays, like many ChIP assays, right? Because, you remove, the day-to-day, handling variability, the sonication, or digestion variability between, this sample, that sample, the cell culture variability of I had to culture, 5 billion cells. Obviously, I'm doing them in batches. The humidity is slightly different this day than that day. I mean, all these little things that, we often take for granted, but really can, impact the overall quality. And that's, of course, not even counting, just handling variability of the process itself and other technical aspects. So the fact that, all of that's happening together with the same materials, right, of course, reduces those levels of variability and allows us to more directly compare proteins to each other. Nonetheless, of course, there are other sources of variability that remain that, for example, differences in antibody affinity, protein types, all the rest that, this alone doesn't address, right? If you have, even for the same protein, two antibodies that have different affinity, you'll have differences in the profiles that can affect that. But at least at the level of a lot of the technical variability, the ability to do them together really does reduce a lot of that noise. But a lot of those types of the antibody specific types of variability, for example, or protein specific variability, those are the kinds of things that one can normalize internally, for example, right, by comparing within proteins within the same experiment.
Yeah. So I'm out of questions. We have hit the hour mark, so that's good. Do you have anything that we might have missed?
Not that I can think of. I mean, I guess the only, additional point I would make, and I sort of alluded to this earlier, is, that I think about ChIP-dip as sort of, one piece of a larger puzzle of really a larger toolkit for multi-omic, if you will, data integration, but and basically of democratizing data integration across many protein types, DNA localization, and also 3D mapping, single cell. And along those lines, I would say that a few months ago, we also published a paper in Cell with my collaborator and colleague at Columbia, Marko Jovanović's lab, developing a method that's very similar to ChIP-dip, but for mapping, for example, many different RNA binding proteins to RNA, a method we call SPIDER. And so while each of these, individual approaches are, of course, valuable for different, we'll say, different types of questions, maybe even different labs thinking about different types of problems in biology, there are also, I think, really great opportunities for integration and for, really thinking holistically about many of the different aspects of, we'll just say, the cell, nucleus of regulation, of gene regulation more broadly. And that's where I think that's, one point that I would want to highlight is for those listeners who, may think beyond even just what's happening at the level of chromatin, but, how chromatin integrates more generally in the context of gene regulation, that there are, really whole frameworks that, one can think about for starting to explore these in different ways.
So maybe coming back to the antibody panels again, because if somebody would be new to epigenetics, right, they have like other data that they have, whatever it might be, and they think that epigenetics would be a key player in their disease, whatever it might be. So would you think that then those panels could be helpful to help them, right, getting started in epigenetics, right? Because if you say, I'm a cancer biologist, and I think now epigenetics playing a role in my cancer.
Is well established, yep.
If you, yeah.
Absolutely.
Of course. But now they're going into epigenetics, and having like a cancer tool set would be like a good thing for them then to see, well, I know that I don't know my factor yet, but it might be that it is in the toolkit then.
Absolutely. And I think having such panels that people can buy, if you will, off the shelf and utilize will be really valuable. And, honestly, the way my dream when we first started this wasn't that every lab would, buy up 100 antibodies or even 20, 30, but really that there would be, commercial suppliers who would have well-validated, high-quality panels that people could just purchase and buy and use like they would any other assay, like they do an RNA-seq experiment, like they do a cut and tag experiment right now, right? They buy a kit, they buy, the reagents. They don't need to be experts in cut and tag to do cut and tag. They don't need to be experts in RNA-seq to do RNA-seq, but they can buy a reagent that's well-validated and utilize it and generate a data set. And if we can do that, right, and if there's a commercial entity that can sell, for example, predefined panels of interest, chromatin panels, cancer epigenetics panels, for example, can even be more specialized, one might imagine, right? That people who aren't experts in this space can now use to augment their understanding. I think that will be, the real transformative potential of methods like this, right? In bringing people from outside the field in, right? And making it easy for people who don't think about chromatin day in, day out to really integrate this into their understanding.
So thank you, Mitch, for your time and for being on the show.
Thank you.
Thanks for listening to this episode of the Epigenetics Podcast from Active Motif. We hope you enjoyed it. You can find all the mentioned references in the show notes. Please rate, review, and subscribe to our podcast on your favorite podcast platform so you never miss an episode. We'd love to hear from you, so please send us your feedback on Twitter, Facebook, LinkedIn, or via email at podcast@activemotif.com, and we'll give you a shout out in a future episode. For more great epigenetics content, check out the Active Motif blog at activemotif.com/blog. Thanks for listening, and stay tuned.